In this third and final episode, we will be covering genetic strategies. Let's start off by talking about the Arabidopsis genome. It's the smallest known plant genome with only 125 megabase pairs. It's the least repetitive sequence of any known higher plant. And it's special because Agrobacterium can easily transform Arabidopsis, which is a prerequisite for many genetic experiments. Agrobacterium-mediated plant transformation is when scientists transfer a section of DNA from bacteria into the plant genome. It starts when a tDNA plasmid is removed from bacterium. This plasmid is cut using a restriction enzyme, and then a piece of foreign DNA is cut with the same restriction enzyme and inserted into the plasmid. You're left with a recombinant plasmid that has this inserted gene. Scientists then reinsert this recombinant plasmid into the bacterium, and it is incorporated into the plant cell chromosome after inserting it into the plant cell. These plant cells are then cultured, and a transgenic plant expressing the inserted gene is generated. This is a useful genetic strategy because it allows plants to safely foster and pass a specific gene researchers are interested in much more quickly than the plant breeding method that is traditionally used. Agrobacterium-mediated plant transformations are inexpensive and a convenient way to perform genetic transformation. Forward genetics and reverse genetics. Forward genetics is the traditional method, or the original method. Its goal is to identify gene or genes that are in charge of the phenotype of an organism. It starts with a mutant phenotype. Scientists then analyze the gene causing the phenotype and finish by cloning the gene or the sequence to establish its DNA sequence. This results in unbiased results because the phenotype is the main level of assessment in terms of gene identification. When plants possess specific phenotypic traits and scientists identify the mutation causing the phenotype, they can then clone this to further understand the function of genes and produce a specific genetic marker. Forward genetics starts with a known phenotype of interest. For example, if a lab is interested in determining the genetics of seed size, a forward genetics approach would be mutating seeds and screening for plants that produce larger seeds. Next, the genome would be sequenced in order to determine what gene or genes the mutation landed in. Both forward and reverse genetics require genetic complementation to establish gene dependency. Reverse genetics is the latest method. A reverse genetics approach starts with a known genotype of interest and uses mutations to link this genotype to a phenotype. This is the latest method. A reverse genetics approach starts with a known genotype of interest and uses mutations to link this genotype to a phenotype. This is only possible because of DNA recombinant technology, which uses enzymes to cut and paste different DNA sequences of interest. These newly cut and paste sequences can be put into vectors that carry DNA into a compatible host cell that then copies or expresses it. For example, DNA may be modified in such a way that it can be replicated in bacteria or yeast. They do this by attaching relevant components to a fragment of DNA and then transferring it into a bacterial or yeast cell, with those elements telling the bacterial or yeast cell to duplicate this DNA alongside its own DNA. This is called DNA cloning, and the cloned DNA that results is sometimes referred to as recombinant DNA. Overall, reverse genetics starts with a protein or DNA for which a function has not been established, and then works backwards to create a mutant gene that in turn creates a mutant phenotype. The goal is to understand the function of a specific gene by observing the phenotypic effects after scientists engineer a specific gene sequence. It examines an organism's phenotype after a known gene is disrupted. To do this, researchers use techniques such as Agrobacterium, X-rays, UV radiation, and chemical therapy to create random mutations in an organism. Another method is called gene knockout. This is when scientists genetically engineer an organism to lack a specific gene or genes. CRISPR-Cas9 is an interesting technology that allows geneticists and researchers to edit parts of the genome by removing, adding, or altering sections of the DNA sequence. As you can see in this figure, researchers edit a genome using the CRISPR-Cas9 as a sort of genetic scissors. They artificially construct a single guide RNA that matches the DNA code where the cut is to be made. The scissor protein is called Cas9, and it forms a complex with this single guide RNA and directs the scissors to the precise location in the genome where the cut is to be made. Using this, researchers can create guide RNAs to localize Cas9 to specific regions of the genome in order to knock out genes of interest. Finally, resources for Arabidopsis researchers. The Arabidopsis Information Resource was founded in 1999 using a grant from the National Science Foundation. It's a database of genetic and molecular biology data for Arabidopsis thaliana. It includes complete genome sequences, gene structures, gene product information, gene expression, DNA and seed stocks, genome maps, genetic and physical markers, publications, and information about the Arabidopsis research community. There's also something called the Arabidopsis Biological Resource Center. This was founded in 1991 by the National Science Foundation and has a goal to represent a central repository for Arabidopsis research resources. They hope to acquire resources from leaders in the field and maintain them by using the best practices. Their goal is to make resources available to researchers and educators around the world.